the next step, that is step 3rd in protein synthesis, is of elongation. Now, where did we leave initiation? At what stage did we say that initiation is completed? If you remember, this was mRNA supposedly. The ribosome's larger and smaller subunit are going to move on the messenger RNA in the direction 5:3. The smaller subunit lies over here and the larger subunit lies over here. This was the P site and this was the A site. A site was empty, ready to take up the amino acid or the charged tRNA according to the coding sequence which was present. Now, as soon as the tRNA comes over here, like this be the tRNA, I am just drawing it like this, let this be the tRNA. When it comes over here, what happens is this initiator tRNA and its amino acid, they come in contact with each other. There is a peptide bond formation by the agency of 23S rRNA. It catalyzes it. It is present in the larger subunit. So, this part of ribosome catalyzes the formation of what? Peptide bond between two amino acids. Alright. Now, this peptide bond stays, this amino acid stays over here. It at the same time catalyzes the reaction where transfer RNA breaks the bond between the amino acid. So, the initiator transfer RNA first of all binds the amino acid with peptide bond with the amino acid that is present in the RNA of A site. One peptide bond is formed and at the same time it breaks the bond that was present between the amino acid. Of course, this would be methionine and the tRNA, and it goes from here. It replaces, it leaves the initiator tRNA, goes from here. What happens is in the next step, this A site is again emptied and this particular amino acid or this particular charged tRNA shifts to P site. So, there is shifting from A to P site, and where has that P site substance gone? It has left it, leaving behind the amino acid which is joined by peptide bond. As the ribosome moves further and further, again next amino acid or next tRNA comes at A site and P site is occupied by the one that was at A site. So, what you are getting after this is that there is a long polypeptide chain that has been formed after many tRNAs have come over here. Alright, this is again one peptide bond. This is again one peptide bond that is being formed. The tRNA has peptides joined over here, 1, 2, 3, 4, 5. The shifting is going on between A site and P site and the polypeptide chain is extending. It is just like supposedly I am standing here, someone comes and joins over here, we join our head, then my body is cut off and the person stays over here with two heads. Then that person shifts, third person comes, then his body is cut off, then the person staying over here has three heads and it goes on increasing. What goes on increasing? The polypeptide chain is formed because every time the next amino acid comes, with the help of this ribosomal RNA, a peptide bond is formed. So, this is how elongation takes place further and further and further. There would be many and many peptide bonds which would be formed. There would be sites. At site new tRNA would be coming, and what we will be having? We will be having a long polypeptide chain that would be formed. Here there would be one tRNA, there would be one tRNA attached to that, there would be the amino acids and they would be attached to the polypeptide and shifting would go on. This is simplest and shabby representation, but I hope you understand what I am trying to tell you. You can see colorful diagrams in your textbooks if you want to, but this is the concept how the polypeptide chain is getting elongated according to the sequence that is present on mRNA as the ribosome moves on the messenger RNA in 5:3 direction. Okay. When this process is going on, you have to remember that there are many elongation factors which are into working and enable this process to be carried out smoothly. Okay, they are known as EFs. There are a few EFs in prokaryotes and many complex ones in the eukaryotes, which is bound to happen because eukaryotes have a complex organization. And with this we come to the end of elongation. The next topic would be termination, where the polypeptide chain formation would come to a halt. How it takes place? Let's see in the next.